Dear student, I am Dr. Prakash Mungli, Professor of Biochemistry. So in this video, I will be going over a polyol pathway. Basically, a polyol pathway is a conversion of excess glucose into sorbitol, and this sorbitol will lead to certain metabolic changes in different tissues and giving rise to macrovascular and microvascular complications, especially in uncontrolled diabetes mellitus patient. Now let's get into what all the enzymes that will convert excess glucose into sorbitol, and that is what we are going to see in polyol pathway. Now, whenever excess glucose is present in the cell, although glucose is converted into glucose 6-phosphate and undergoing glycolysis in most of the cells, but whenever there is a buildup of glucose, slowly some of the glucose can go into other alternate metabolic fate, and this specially happens in a tissue that is insulin independent, because insulin is the one that will regulate the entry of glucose into the cell in certain tissues like skeletal muscle, adipose tissue and cardiac tissue. But in insulin independent cells, like all other cells other than skeletal muscle, cardiac tissue and adipose tissue, you don't need insulin to get glucose inside, because those are insulin independent cells. They have a glucose transporters which will work based on the concentration gradient. That means as long as there is more glucose in the blood, so glucose is taken into the cell. So that means more than necessary glucose is getting into these insulin independent cells, especially if a person has got hyperglycemia. So, and when a person gets into hyperglycemia, it can be a pre-diabetes condition, it can be a diabetes condition which is not under control. So uncontrolled diabetes mellitus, or constantly being present in well-fed state, especially on carbohydrate diet. So excess glucose level in the blood, which leads to increased gradient there in the blood stream, like glucose elevated in the blood, hyperglycemia, allowing more and more glucose to get into insulin independent cells. So what will happen to this glucose? Although some of the glucose can go into glycolysis, but that extra glucose which is there, it will be diverted into a molecule called sorbitol formation. And the sorbitol formation is done by an enzyme called aldose reductase enzyme. This aldose reductase enzyme is a general enzyme. It is going to act on all aldoses, aldohexose in fact, like glucose, galactose. Okay, so glucose is converted into sorbitol. Likewise, galactose is converted into galactitol, but glucose is the more predominant molecule, monosaccharide in our blood than the galactose, right? So the glucose, excess glucose in the cell, because of the increased activity of aldose reductase, it will be converted into sorbitol, and that sorbitol is an osmotically active molecule. And this sorbitol, which is going to imbibe water inside the cell, and more water gains access to the cell because of osmotic activity of sorbitol, and that's why cell will swell. And when the cell swells, it leads to metabolic changes in that particular cell, and sometimes because of the swelling of the cell, it may even break down. Okay, and that's how excess glucose can affect cellular metabolism by simply allowing water to get in because of its osmotic activity. Now, what all the tissues where aldose reductase activity seen at higher level? So some of the tissues like lens, retina, squamous cells of peripheral nerves, liver, kidney, placenta, red blood cell and cells of ovaries and seminal vesicles, they all will show increased activity of aldose reductase enzyme. That means all these cells are able to convert more and more glucose into sorbitol only when glucose is available at high concentration. Why? Because aldose reductase enzyme is a high Km enzyme. it has got high Km for glucose. That means what? It has lower affinity for glucose. That means only when glucose levels are kind of rising in the cell and that glucose is converted into sorbitol. So only when it is present at higher concentration, because aldose reductase is a high Km enzyme, that means it has got lower affinity. So point that you need to remember is increase in aldose reductase enzyme activity will lead to increase in sorbitol, and increased sorbitol means increased osmotic activity, and increased osmotic activity means abnormality in the cellular metabolism. Just remember that fact. Now, certain tissues, they have got a mechanism to handle that sorbitol. Now, as you see here in this particular diagram, so we have glucose which is an aldose sugar, aldehyde group is shown there, and that glucose is the aldehyde group of glucose by aldose reductase enzyme is converted to alcohol group there. Now take a look at all the carbons present in this sorbitol molecule. Right, first carbon has alcohol group, second carbon has alcohol group, third carbon has alcohol group, fourth carbon alcohol, fifth carbon alcohol, sixth carbon alcohol. So all the carbons have got alcohol group. That is why name of this pathway is polyol. Poly means what? Many alcohol groups, right? So that is why the name here is polyol pathway, and the polyol is a sorbitol. So the polyol form of glucose is sorbitol, and only when there is excess glucose in the cell, some of that glucose is diverted into sorbitol, done by increased activity of aldose reductase enzyme, because it's the high Km enzyme for glucose, and also there is a use of NADPH+. So whenever there is excess glucose, some of the glucose diverted into pentose phosphate pathway to provide NADPH. So there will be plenty of NADPH anyway. So that will promote conversion of glucose into sorbitol, and sorbitol will exert a osmotic activity. Now certain cell, and where this happens, it happens in all the cells that I have named in the previous slide. It is some of the cells to mention again: lens in the eyes, retina, kidney, squamous cells of the peripheral neurons, right? So seminal vesicles, ovaries, liver, all they will express increased aldose reductase enzyme activity, making sorbitol. But only thing is, only 3 tissues in our body will express sufficient sorbitol dehydrogenase enzyme, which is a protective enzyme. Let's look into that. Sorbitol dehydrogenase enzyme. So the next enzyme that is shown here, this sorbitol dehydrogenase, it is going to convert sorbitol into fructose. Fructose is not an osmotically active molecule compared to sorbitol. So sorbitol dehydrogenase, it is going to convert sorbitol into fructose, thereby decreasing the osmotic effect of sorbitol. So that means sorbitol dehydrogenase is a good enzyme, because it's going to decrease the concentration of sorbitol. But the thing is, sorbitol dehydrogenase is expressed at high levels only in the liver, ovaries and seminal vesicles. Other tissues, they do not express much of sorbitol dehydrogenase. They express only very low levels of sorbitol dehydrogenase. That's why once the tissues converts glucose into sorbitol, most of the tissues simply accumulate sorbitol except liver, ovaries and seminal vesicles. That's why increased aldose reductase enzyme activity will lead to major issues in the lens, nerves, kidney and retina, giving rise to, in the lens it will give rise to bilateral cataracts, in the nerves it will give rise to neuropathy, in the kidney it will give rise to nephropathy, and in the retina it will give rise to retinopathy. All these complications of excess glucose is partly explained because of accumulation of sorbitol. And these are the macrovascular complications that are seen in uncontrolled diabetes mellitus patients. Now, sorbitol levels can be decreased in diabetes patients by prescribing a drug which belongs to aldose reductase inhibitor class. Now the aldose reductase enzyme can be inhibited by certain drugs, because these drugs, what they do is they are going to inhibit this aldose reductase enzyme, thereby decreases the conversion of glucose into sorbitol. Just to name a few names here, it is sorbinol. Sorbinil is an aldose reductase inhibitor. Then we have ipelastat, imirastat. Any drug that ends as, just to give you a hint, any drug that ends as stat, okay, the last name as stat, any drug that ends as stat, usually it is a aldose reductase inhibitor. Don't get confused with the statin. Drug that ends as statin, it is HMG-CoA reductase inhibitor. That is in cholesterol metabolism. In hypercholesterolemia, we use the drugs that ends as statin. Whereas any drug that ends as stat, like as it is shown here, ipelastat, imirastat, right? Look for the stat at the end. And those are aldose reductase inhibitor along with sorbinil. Okay. So this is what is a polyol pathway, which involves conversion of glucose into sorbitol. So that's about polyol pathway. I hope this particular short video has helped you in understanding polyol pathway and also a clinical application of polyol pathway in understanding why do we see complications in uncontrolled diabetes mellitus patients. Thank you.